Assalamualaikum and good day. My name is HANISHA BINTI HAIRUDDIN. My final year project is titled Investigating Transmembrane Proteins as Novel Drug Targets in Leptospira interrogans.
So this is the outline of my video presentation.
First, the background. Leptospirosis is a major but neglected global disease causing over 1 million cases and 60,000 deaths annually, primarily in the tropical region. It is a zoonotic disease transmitted from animals to humans through water or soil contaminated with infected urine. Infection can progress from mild flu-like symptoms to severe and fatal organ failure. The problem is clear. It is a widespread deadly disease with minimal treatment options, and current laboratory methods for discovering new drug targets are slow and inefficient. Crucially, a comprehensive computational analysis to find these targets in Leptospira is lacking. Our research addresses this gap. We use bioinformatics to scan the pathogen's entire proteome, aiming to identify novel target candidates and establish a foundation for future drug and vaccine development. Our research has two main objectives. The first one, to identify transmembrane proteins in the pathogen Leptospira interrogans. Second, to evaluate the potential of these identified proteins as drug or vaccine targets. The significance of this work is fourfold. The first one, it bridges a gap. It turns the unused genetic data into practical biology clues. Second, it aims for impact. The goal is to find new targets for much-needed vaccines and diagnostics. Third, it saves time and resources. It's a cost-effective way to pinpoint the most promising leads for future experiments. Fourth, it demonstrates values. It shows how a student project can meaningfully contribute to fighting a neglected global disease. The existing literature reveals a consensus on the importance of surface structures, but also a critical gap. Studies by Samrat et al. And Bolet et al. Established that outer membrane proteins are fundamental for host cell adhesion and invasion. Furthermore, Sun et al. And Philip et al. Demonstrated that surface components triggers a severe inflammatory cytokine storm. However, a pivotal study by Xu and Yang provided a crucial distinction. They found that while some proteins are individually essential, others are not due to functional redundancy within the bacterium's proteome. This means a protein can be replaced, making it a poor drug target. This insight underscores the necessity of our work, a broad, systematic research for essential, non-replaceable targets, moving beyond studying only the known virulence factors. This diagram shows the membrane components. This schematic details the pathogen's complex outer envelope. It highlights key structures, including the outer membrane beta-barrier proteins and lipoproteins, which are directly involved in interacting with the host environment and are primary candidates for therapeutic intervention. This diagram shows the transmission cycle. This illustration depicts the zoonotic life cycle of Leptospira. The bacteria persist in the kidneys of reservoir hosts, such as rodents, and are shed into the environment via urine. Humans and other incidental hosts then become infected through direct contact with contaminated water or soil, completing the transmission chain and highlighting the critical environmental component of this disease. This body of literature also details the severe organ-specific pathology, justifying the need for targeted therapy. In the lungs, research by Nicodemo and Duarto Neto shows damage results from both direct bacterial invasion and a dysregulated immune response, culminating in the severe pulmonary hemorrhage described by Chalkajamwat et al. Similarly, in the kidneys, work by Kumar et al. And Shou et al. Establishes that acute injury arises from this same combination of direct toxicity and immune damage. Crucially, later studies by Fernandez et al., Lee et al., Wu et al., and Noh and Padanilam have begun to molecularly define these processes, identifying specific bacterial factors that drive tissue damage. This progression from clinical observation to mechanistic understanding pinpoints the molecular events that new antimicrobial agent could disrupt. This brings us to the core gap. Historically, research has focused on a limited set of proteins like LIP-L32. Consequently, a comprehensive inventory of all potential therapeutic targets, especially among transmembrane proteins, remains unavailable. To date, the entire bacterial proteome has not been systematically screened using bioinformatics to identify novel essential targets that lack functional redundancy. Our project is designed to fill this significant gap. To do this, there are several step-by-step bioinformatics pipeline. The first one, data sourcing and filtering. We began with the UniProt KB database to obtain the complete verified proteome of Leptospira interrogans and filtered for larger, more functionally significant proteins. Second, transmembrane prediction. We use the tool TM-HMM to identify which of these proteins are embedded in the bacterial membrane, which are a crucial location for drug action. The third one, sequence and feature analysis. We then use BLASTP to compare these membrane proteins against all known proteins, searching for unique bacterial sequences that could be targeted without harming human cells. Finally, we use ProtParam to calculate basic chemical properties to assess their druggability. In summary, this method filters thousands of proteins down to a specific data-driven shortlist of targetable membrane proteins. The expected outcomes. We predict the MHMM will identify multiple transmembrane proteins. We will characterize their topology and generate a prioritized shortlist. We will prioritize uncharacterized proteins with high conservation across pathogenic species as potential drug targets and proteins with cleavable signal peptides as potential vaccine candidates. The limitations and mitigation. We acknowledge the limitations, which are reliance on prediction without lab validation, database dependence, and a narrow focus on transmembrane proteins. Our mitigation strategies include using multiple bioinformatic tools for cross-validation, using the latest databases, and most importantly, clearly stating our findings as testable hypotheses. We will explicitly recommend essential feature wet lab work such as knockout studies, protein expression, and animal models to validate our computational discoveries. This Gantt chart outlines our projected timeline. For the next semester, I will meet my supervisor to finalize our research plan and begin executing the bioinformatics pipeline. That's all from me. Thank you for your time.